Hello, everybody. Welcome to Ms. McGuire online video lecture. And today we're covering DNA replication. Now, this chapter was divided into two video lectures. In the first lecture, I talked about the discovery of DNA as a hereditary material and structure of DNA. In this video, we will focus on DNA replication. But before we begin, I want to go back to the first slide and remind you that that genetic material must carry out two jobs: duplicate itself and control the development of the rest of the cell, right? So DNA, if we're talking about genetic material, what can be that molecule that can regulate activities of a cell, right? And at the same time, has the ability to make a copy of itself, right? So it can be passed to next generation of cells, and as a result, to next generation of living organisms. And the only molecule that satisfies these two requirements is DNA. And that's why it's very important that DNA can replicate, right? So let's go ahead and begin. Now, as soon as Watson and Crick describe the structure of DNA, if you don't know about Watson and Crick, please watch my first video on this topic, where I pretty much explain who those guys were and how they describe the structure of DNA. But after they figure out how DNA is made, its mechanism of replication becomes obvious. Now, Watson and Crick get a Nobel Prize for the discovery of DNA structure. And of course, they published their findings and their work. And at the end of their report on the structure of DNA, there was a statement. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible coping mechanism for the genetic material. So by understanding the structure of DNA, they made a proposal. They hypothesized the mechanism of DNA replication. And of course, before it was accepted, the mechanism of DNA replication was accepted, scientists conducted many experiments that support the claim, right, or the hypothesis that DNA replicates as today we know semi-conservative mechanism, right? So I guess, I'm sorry, it might be a little bit complicated. What I'm trying to say over here, that structure really tell us a lot about function, right? And if we know structure of DNA, we can suggest the mechanism for its replication just based on how those nucleotides are arranged within a DNA molecule. So let's see what all this means. Over here, okay. So Watson and Crick envisioned DNA replication to be semi-conservative. When two identical double helices are formed from one original parietal double helix. So each new DNA helix serves half of its original DNA. And we're going to look at this mechanism first, and then we will go back to this experiment of Mendelssohn and Stoll. So here we have a DNA, right? So that's our original DNA. We're gonna start with this DNA, and our goal is to make from one DNA to make two, right? To make identical copies of this original parental DNA. And if you see how this parental DNA has this like dark blue color, and if you look at the daughter DNA, you can see that each daughter DNA has one strand of old DNA and one strand that is new. That means, so that's what, this is what that previous slide told us, that the two identical double helices are formed from one original parietal double helix. So we started with one original, right? So here's, and let's just, let's say we have, it's our original DNA, double helix. Now each new DNA will have half of the old DNA, here half of the old DNA, and half of the new DNA. So that says each new DNA double helix, so this is our new DNA double helix, and this is our new DNA double helix. It conserves, so it keeps half of the original helices. You see this blue part? That's our original helix. So in new DNA, you have half new and half old, right? Okay. So now Matthew Meselson and Franklin Stoll in 1957, they were American geneticists. They demonstrated the semi-conservative mechanism of DNA replication with a series of density shift experiments. They labeled the parietal DNA with heavy nitrogen, and they showed that those high-density nitrogen was incorporated into one strand of each daughter double helix. So labeled replicating DNA from bacteria with a heavy form of nitrogen and traced its pattern of distribution. So, and this is what they did. So they took the parietal DNA, right? Yeah, then they label it by heavy nitrogen. So the heavy chain shows in dark blue. And then they trace this heavy nitrogen in a daughter cell. And they show that each daughter, not daughter cell, daughter DNA, each daughter DNA have half of the DNA of parent with this heavy nitrogen. So that's what semi-conservative is. Okay, steps in DNA replication. DNA replication occurs during the S phase of the cell cycle prior to cell division. Now here you can see cell cycle, that is a series of events from the point when cell is formed and then cell divides, right? So when cell is formed, it goes through several stages like G1, S, and G2. Those parts of interphase. And at the end of interphase, cell divides, and this is called mitosis. Now, in mitosis, cells divide, but replication of DNA happens prior to it in the S phase, and S stands for synthesis. So it's prior to replication in the interphase in S phase. When DNA is replicated, it separates and hydrogen bonds holding the base pair together break. Two identical nucleotide chains are built from one as the bases form pairs. A site where DNA is locally opened is called replication fork. Now let's just kind of try to understand here why we even need to replicate DNA. Now look, if you have a cell, and this cell has DNA. Now, if you want to make two identical daughter cells, right, this cell must divide. Each cell must have the same DNA, right, to be identical. Now, if this cell divides, we don't have the cell anymore. We have two daughter cells, but each one has the same DNA. What does it mean? It means that first we need to make a copy of DNA so you can spread this DNA between two daughter cells, right? That's why we need DNA replication, right? And we're looking at DNA replication here. So first, we have our parent DNA molecule, right? This is number one, right here. Parental strands unwind. So number two, you see how we unwind them and separate at several points. That's number two. Each parental strand, shown here in dark blue, provides a template for DNA polymerase, and that's a special enzyme, to bind complementary bases, A with T and C with G. Now look, when you have DNA and when you open it up, unwind it, you see those bases here? And it says G, A, C, and T. When you open up the DNA, you have enzymes that are gonna bring complementary bases to existing bases. And we know if we have G over here, then we're gonna pair C. If we have A, we're gonna pair T. So those bases shown in the light yellow or light blue color, that's a new strand, right? And those bases are added by special enzyme called DNA polymerase. Then sugar phosphate backbone of daughter strand closes, right? So that's between complementary bases, we will have hydrogen bonds, and here we close these strands and we're forming two identical DNA molecules. Now, to create DNA, to replicate DNA, we need many different enzymes. So here's the enzymes that are involved, some of the enzymes that are involved. I would say most important, but we have other enzymes, and I really hate this word, most important. It's just some of the major enzymes, let's put it this way, and then we will look at function of those enzymes and how exactly they work. So the first enzyme here is helicase that will unwind parallel double helix. So over here, this purple oval structure represent enzyme helicase, and we see how we unwind or open up DNA molecule. We need binding proteins right here that stabilize separate strands, because otherwise they have tendency to close up. So we need them to stay open. Primase gonna add short primer to template strand. So here's a primase, and it's gonna add just a primer, kind of like from this primer we start synthesis, right? But that's a primase. DNA polymerase that's gonna bind nucleotides to form new strand. This is now we forming double helix. And ligase is the enzymes that will join what it's called Okazaki fragments and seals other nicks in sugar phosphate backbone. Now, keep this in mind, these Okazaki fragments. So for now, we're gonna skip this part for a minute, and then we will get back to these Okazaki fragments. And why do we have them, and why do we need them? All right, so let's start the process. First, helicase binds to origin and separates strands. So here's our helicase, and gonna bind to a specific part of DNA and open up two strands. Binding proteins keep strands apart. So here's our binding proteins. Primase makes a short stretch of RNA. Remember, we're making DNA, but we cannot start synthesizing DNA from nothing. We need a primer, like a beginning of the strand. And this primer gonna be short molecule of RNA. All right, so here's our primase, and it's put these little fragments of nucleotides. It's a short RNA that will be our primers. Now look what is important over here. Remember, DNA is double helix, and we have two strands, right? One strand over here and another strand. And those strands are anti-parallel. That means if one strand runs from 5 to 3 direction, and 5 is from phosphate group to sugar, 3 is sugar. So we go phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. That another one is anti-parallel. So it's going to be sugar, phosphate, sugar, phosphate. So here we have sugar, and here we have phosphate. However, so you see how 5 kind of 5 and goes with 3 end, right? So when we separate, here we have our 3 end, and it's need to go with 5 end. And this is our 5 end need to go with 3 end. That's why those little primers are also going to be put in a opposite direction. So this one put from 5 to 3 this way, and this put in a 5 to 3 in the opposite way. See how this in 5 to 3, 5 to 3. And now where we're gonna add DNA nucleotides? Are we gonna add, because look, primer shown here in green, but we need to add this blue parts that are DNA nucleotides. Are we gonna add it to 3 end or to 5 end? To 5 end over here or to 3 end? Well, actually, these nucleotides can be only added to 3' end. So you can only add nucleotides in one direction. So you can synthesize new strand from 5 to 3 only. Now over here, you start adding nucleotides in the opposite side, right? Now let me get a pen. So here you're gonna add nucleotides. So you're gonna add here, 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 and here, right? And you kind of continue. Here you see I'm building the second strand, right? So you're going this direction, and this is a direction where a helicase moves, because this is a helicase, it moves here in a direction from 3 to 5. So it will open up, and you're just gonna add fragments. So this is called leading strand. But on this strand, you cannot start adding nucleotides to a 5 end. You're gonna add them right here, right? So you add them in the opposite way. And here you have primer. You cannot add anything on this side, right? So you see how kind of like it doesn't make sense. So pretty much what do you need to do? You need to put a new primer here. Let me put the green one, right? You need to put a new primer here, again, green, right? And then you start adding, again, on this side, 1, 2, 3, 4. Kind of you synthesize this lagging strand in fragments. So here's a fragment 1, here's a fragment 2, and what you have between them, you have that primer. Now those fragments are called Okazaki fragments. And it's a little bit more difficult, right, to make the lagging strand, because not only you cannot synthesize in a continuous pattern, you also have these primers in between. So you need to remove those primers, and you need to seal those fragments together. So it seems like more work, right? And that's exactly how things happen, right? So it shows what I just showed you. See how this in a leading strand, you just pretty much continue synthesizing along the molecule, the whole molecule of DNA, and on the lagging strand, you're synthesizing in the opposite direction. So DNA polymerase adds DNA nucleotides to RNA primer. So you see how we added over here, where we have 3', we added the blue part, and this way we have a 3', we added the blue part. That's our DNA nucleotides. So they can only be added where we have our sugar, right, to 3' end. DNA polymerase proofreading at the same time when it's adding nucleotides, it's a proofreading the new strand to check and replace incorrect bases. Right, so that's so far so good, right? Continuous strand synthesized continuously in a 5 to 3 direction. Right, so again, that's our 5. This is our 3, and you can always add more to the 3 end. So it goes from 5 to 3, and now look, everything looks good. 3 with 5, the same as here. We have 3 with 5, so we have still anti-parallel strands. So we continue, it's continuous synthesis. And we have discontinuous synthesis that produces Okazaki fragment on a 5 to 3 template. So over here, we again, because it's our 5, right? So over here, we cannot move along, we actually moving backwards into short fragments that called Okazaki fragment. And each Okazaki fragment has a primer and a part of a newly synthesized DNA. That makes sense? And if it doesn't make sense, so let's go back here. And when we just put those primers, see how we put primer. We cannot put primer, and get a green one. So over here, because we have 5, we're gonna put primer. We cannot put primer over here that also go from 5 to 3. Right, we cannot put primer like this. I'm gonna cross it out to show you we cannot do it. Because we need to keep it anti-parallel, because we cannot have 5 end and 5 end on the same side. We always need 5 and 3, 5 and 3, right? That's why we cannot put primer this way. We put primer the opposite way. So we have now from 5 to 3 this direction, but we can only add nucleotides to 3, to part 3, right? To 3' end. That's why we kind of like, seems like we're in trouble over here, right? So we can only, so with the leading, it's no problem. We're just gonna synthesize a new daughter strand just continuously. Here, we're gonna synthesize kind of backwards, forming those short fragments that have very, pretty name, Okazaki fragment. Now we need enzymes that remove the RNA primers. That we're gonna remove one RNA primer on a leading land and several on a lagging. And if we have this Okazaki fragment, we also need ligase that seals a sugar phosphate backbone. Ligase seals the sugar phosphate backbone on both strands, and it also will seal Okazaki fragments. All right, so I just hope I did not confuse you too much. Let me kind of make a very quick summary, right? So first of all, how DNA replicates. It's a semi-conservative mechanism. That means that we have a parent DNA that is made of 2 strands, right? Then we will unwind the strands, and we will use each strand as a template for a daughter strand. That means that a new DNA will be half new and half old, right? Then we're gonna use many different enzymes. The first enzyme, that helicase, will unwind the DNA. Binding proteins will keep it open. Then we need a primase to put a primers, because we need to start adding nucleotides to a primer only. And DNA, because it's made of 2 strands, one strand called continuous strand, or leading strand, and it's going to be synthesized continuously. We're going to put a primer, and we're going to just start adding nucleotides on 3 ends. So here one nucleotide, another nucleotide, another nucleotide, and we just keep adding them, and we continue to add. Then, and the enzyme that's going to add those nucleotides called DNA polymerase. On a second strand that is called a lagging strand, we're going to add nucleotides away from a side of the opening. So here we open this way, but we're adding short fragments in the opposite way. Then we will need to add another primer, right? So here we're going to add another primer, and we're going to start adding nucleotides. Again, kind of away from the point of DNA where we open it up. So we're going away. So we're forming what is called Okazaki fragments. Then all these nucleotides and the fragments need to be sealed together. Primers need to be removed. And that's a job of another enzyme called ligase. Right, so ligase is going to add all these nucleotides together, forming the sugar phosphate base, and it will add Okazaki fragments together. And we will be done right here. Now, because DNA is a huge molecule, if we start replication in one point, and we need then we need long time to make a copy. That's why we're gonna start replication into a different into many different places at the same time. And it's so the sites of replication resembles bubbles that coalesce together as the daughter double helix is formed, right? So origin of replication, we have several origins of replication on a parent DNA, and we're gonna start like in many places at the same time. We form these replication bubbles, and then we'll form our 2 daughter DNAs, right? So kind of like instead of, okay, so here's my, let's say here's my DNA molecule, right? Double-stranded DNAs, let's remind us a letter. And we're gonna start replication. We're gonna start replication, let's say here and here and here along the whole length of DNA, and then the process will speed up. Okay, so now we know the DNA can replicate itself, so that's good. So DNA can make a copy. These copies can be passed to new cells. Now, now imagine we need to kind of look at somebody's DNA molecule. Let's say there was some crime committed, and we have a couple cells left from the from the person who committed crime. It's just a few cells, and they have very small amount of DNA, and we want to analyze this DNA. Now, if you have so small sample of DNA, it would be good if we have some amplification technique. Can we make from one DNA, for example, 10,000 copies? Can we do it? Or 10 billion copies? So actually we can, and that's the process called polymerase chain reaction, or PCR. It's a DNA amplification technique. So what it means, if you have like one DNA or one fragment of DNA, you can make many copies. So it uses DNA polymerase, that enzyme that we just described, that polymerize, that make new DNA. So it uses this DNA polymerase to rapidly replicate a specific DNA sequence in a test tube. It yields more than 10 billion copies of the target DNA sequence after 30 cycles. So now imagine if we start with one DNA, and after 30 cycles only, we will have 10 billion copies. Now we have plenty of genetic material to do our, whatever analysis we need to do. It's useful in forensic investigation to amplify small DNA samples that we just described. So how is it exactly done? So here, for example, we have DNA, and we have target sequence, or this, in this situation, it's we have a virus genome, and we have a specific part of DNA we're interested in, or it can be a human DNA. But we need this piece of DNA, we need primers, we need free nucleotides, because we're gonna make a copy of DNA from nucleotides, and we need polymerase, DNA polymerase. But in this situation, we need heat-resistant polymerase. So if we mix all this together, we're gonna apply heat to unwind the DNA. In a living cell, we have enzyme called helicase. But in a PCR, we don't have this enzyme. We just use heat to unwind the DNA. And that's why we need heat-resistant DNA polymerase. Right? So when we mix everything together and we open up DNA, those primers, because we have primers to begin with, they will primers hydrolyze, they will attach to DNA due to base complementarity. So that spontaneously will happen. And then our DNA polymerase start adding nucleotides to old DNA strand. And we will repeat it, process many times, that give us amplification. So we use the knowledge about DNA replication now to replicate DNA in a test tube and make many copies when we need it. So cells make copy of DNA only when they need to divide. We can make copies of DNA in a test tube whenever we want to, and we can make billion of copies in just a short period of time. Right. So, and this is just steps of this amplification of DNA using PCR. So we select target sequence in a genome. It can be viral genome or any genome. We need to have primers, free nucleotides, and heat-resistant polymerase. We're gonna pretty much mix everything together. We're gonna add we're gonna increase the temperature, and temperature will or heat will separate strands of DNA. Now primers hybridize. Hybridize means they will form the hydrogen bonds with complementary bases. DNA will fill in, and we will repeat this process as many times as we need, and that will give us many copies of original DNA, and this process is called amplification. So PCR has been used to amplify DNA for extinct animals, microorganisms that cannot grow in laboratory, the brain of 7,000 years old human mummy, the digestive tract of carnivores to reveal food webs, roadkills and carcasses washed ashore to identify locally threatened species, genetically altered bacteria that are released in field tests to follow their dispersion, poached moose meat in hamburger, remains of Jesse James grave for positive identification, in intestines of genital crab allies and rape victims, which match the DNA to the suspect. So this is just application of this PCR technique and amplification of DNA. So you can see when we have a small DNA, we can make many of them, and that's when we use this. So this is examples when we use this PCR. Okay, so now we good. Now we know how DNA are replicated. We know that we can, because we know how DNA is replicated, we can speed up this process and perform it in a test tube called DNA amplification. What else we can do? If if we know the process of DNA replication, we can figure out sequences of nucleotide in a DNA molecule. So remember, DNA is made of like C, T, A, G nucleotides, right? So what is our sequence? Is it C, C, T, T, A, G, or is it G, C, A, T? So we can really figure out the sequence of nucleotide and why would we need it. So imagine that you have a healthy person, right? And you look at this healthy person gene for I don't know, for cystic fibrosis, for example. So you have a person who is healthy, he or she doesn't have a disease. So you look at a gene of this person and you know the sequence. And then you have a person who has genetic disease, like maybe cystic fibrosis. And you look at the same gene, and if you know the sequence of nucleotide in a damaged gene, you know where mutation happened. You know what gene is responsible for that specific disease or disorder, right? So it's like I have DNA, you have DNA, but it's like a book that we need to learn how to read. And to read it, we need to know the sequences of nucleotides, right? And we can do this sequencing called Sanger sequencing. So it's sequence individual genes. So you have a gene for blue eye color, right? Or you have, let's say, gene for blood type A. But gene is made of these C, T, A, and G nucleotides. But how exactly, what is their sequence, right? What is their order, right? So when we are trying to figure out what that order is, that's called sequencing. It's also used to check the accuracy of a selected sequenced area of a genome. So that tells us, well, he's a healthy person, and healthy person has sequence CCTTAG GCAT, and that's a normal sequence. Now, if how we can check accuracy, we take another person who has a disease, and now we see that this person has CCTTAG GCA, and maybe instead of T over here, maybe it's a G, and we know how. Look, that's where mutation is, and that mutation causes this particular disease. Okay, now how exactly it's made? Well, this is a little bit it's not that easy to explain in a couple minutes, but I'll just try. Just gonna be overview, okay? Just gonna be very quick overview. How it's done, you have for example, look, let me do. Okay, so here you have a DNA, and let's say this DNA is A, T, C, C, and G, and A again. Right? Now you're gonna add you're gonna add the fragments of DNA in a test tube that will be complementary with what we have. See, let me get a different color over here. So for example, you're gonna add a fragment that is T. So you're pretty much gonna make a copy of the DNA, right? So you're gonna make a copy of one strand. And you know, to make a copy, A pairs with T, T pairs with A, C pairs with G, right? So that's gonna be our so that's kind of a short piece that complementary to the DNA that we have. And the last nucleotide will be the one that you cannot add any more nucleotides to to this last nucleotide, right? So it's going to be like a short fragments, and those short fragments are going to be different lengths. So you see over here, and the those end nucleotides, they will add fluorescent dye to them. And then we can separate them based on how long they are, and we will know what is the sequence of nucleotide in the original DNA. And so then we, for example, over here, we're gonna add now a new, another one, and it's gonna be, for example, just T and A. T and A, right? So that's T and this A, and we cannot add anything to A. So that's, this piece will just, it will be attached to the beginning of this. So we will have obviously several DNA molecules. Let me just remove this and I start all over, because otherwise we don't understand. So here's our DNA. Okay, it's gonna be yellow now. No, I don't want it yellow because I don't want it to be confused. All right, so here's our DNA, and let's say our sequence is A, T, C, and C. All right, so we're gonna add, and okay, so here's it again. So we will have several copies of this DNA. All right. And then we're gonna have the fragments here. One gonna be, let's say A. Oh, I'm sorry. Erase it. So one gonna be T and A. And this is where we will form, right? This is where we will form connection, and nothing can be attached to an A. Another fragment gonna be shorter. Oh, in this example, let's make it longer. So we have T, A, and G, and this is where we're gonna form our connection, and nothing can be added. Nothing can be added to the last one. Right, so we have kind of like, we synthesize the shorter fragments from original DNA, shorter and longer. And how we make sure that we don't make a copy of entire gene, because the last nucleotide is just altered the way that nothing can be attached. So we end up with fragments of different lengths. So DNA fragments are ordered by size on the sequencing gene. So then we will separate them using this special gel, and the smaller fragments will move further down through this gel, and the heavier, longer fragment will stay on at the beginning of the gel. DNA, by the way, it has a charge. DNA is negatively charged. So this fragment will be negatively charged. This one, they're all negatively charged. So negative and positive attaches. So those fragments gonna move from negative over here to positive. But when they're small, they're gonna move further. And the bigger they are, they will be on the top of this gel. So when we have the separation of the fragment, then we can figure out based on the last nucleotide that has a fluorescent dye attached to it, we can figure out the sequence of DNA. Right? So that's how we read a DNA sequence. And of course, it's done automatically now. It's done through the using UV lights shining to that gel, where we just separated fragments of DNA based on their lengths, and then the computer just reads the sequence of C's, T, A, and G's in a molecule of DNA. And when we know the sequence, we know how normal gene look like, and we know how the mutated gene look like. Right, so let's just summarize over here very quickly. Now we discover the structure of DNA. We know that DNA is the molecule of inheritance. And because it's a molecule of inheritance, it must replicate. It must be able to make copy of itself. And we know that this is semi-conservative mechanism. And by knowing how DNA replicates, first of all, we can use PCR to make many copies. If we already know how the copies are made, then we can make these copies in a test tube, and we can have billions of copies of DNA to study them, right? For example, in forensics, when we have just a small amount of DNA, we can multiply it, amplify it, have a billion of them, so we have more genetic material to work with. Plus, if we know how DNA is replicated, we can use this knowledge to separate DNA into fragments of different lengths. And by doing it, we can figure out the sequence of nucleotides in a DNA. And it's very important to study sequence of DNA nucleotide so you can compare a normal gene with a gene that has a mutation. Okay, I hope it does make sense. Thank you for watching, guys. And if you have any questions, let me know. I know it's might be a little bit difficult. If this is the first time you're hearing about those processes, you might be kind of like you might be confused and like, oh, how this stuff works. But if you rewatch the video several times, then you hopefully will understand it better. So anyway, thank you for watching, and I talk to you next time.